In the name of Allah, the most gracious, the most merciful. Assalamu alaikum. Today we will discuss about linkage map, 3-point test cross and interference. So let's start. What is linkage map? Linkage map, also called a genetic map. It shows the relative positions of genes on a chromosome and also helps to visualize the arrangement of genes and the distance between the genes in terms of genetic recombination. How to create a linkage map? There are three main steps involved in the creation of linkage map. First one is to perform a genetic cross. Cross the organisms that are heterozygous for the genes being studied, and also analyze the offspring to determine how often recombination occurs between the genes. The second step is the calculation of the recombination frequencies. So what is the recombination frequency, how to calculate, and what is the unit of recombination frequency? All these topics we have already discussed in our previous lectures. And third step is the arrange the genes in linear order based on recombination distances. Let's have an example of linkage map. Suppose we have three genes A, B, and C. After a genetic cross, we find gene A and gene C are 8 centimorgan apart from each other. Gene B and C are 3 centimorgan apart from each other. And gene A and B are 11 centimorgan apart from each other, which is the sum of the distances between A to C and B to C. Since A and B are farthest apart and C is between them, so this is the genetic map based on the distances among all the three genes. A 8 centimorgan apart from C, and C 3 centimorgan apart from B, and A 11 centimorgan apart from B. This map shows the relative positions of genes on chromosome. Our next topic is 3-point test cross. A 3-point test cross is an important genetic tool used to map the order and distance between three linked genes on a chromosome and to determine the recombination frequencies between them. What is the cross setup in 3-point test cross? It includes two types of parents. Parent 1 will be heterozygous for all the three genes, and parent 2 will be homozygous recessive for all the three genes. And the first step is the gamete production. Parent 1 produce 8 kind of gametes and parent 2 produce only 1 kind of gametes. The 8 kind of gametes that is produced by the parent 1 have been categorized into 3 main types. Number 1, parental type. Number 2, single crossover type. And number 3, double crossover type. What is the parental type? Parental type is basically non-recombinant. And what is the single crossover type? Normally, a single crossover occurs when there is one recombination event between two of the three linked genes, either between A and B or between B and C. And what is double crossover? A double crossover occurs when two recombination events takes place. One between the first two genes and another between the second two genes. These are the visual representation of single and double crossovers. So first we discuss the single crossover. Three linked genes A, B, and C, and A, B, small b, and small c. And in single crossover, as we already discussed, only one recombination event occurs, either between the first two genes or between the second two genes. In this case, first two genes. So A, small b, and capital C. Capital A, small b, and then capital C. Capital A, small b, and capital C. And in this case, small a, capital B, capital C. Small a, capital B, and capital C. This is the single crossover. And in double crossover, as we mentioned, there will be two recombination events between first two genes and second two genes. Capital A, small b, capital C. Capital A, small b, capital C. Small a, capital B, small c. Small a, capital B, small c. And this is the double crossover event. The second step is the offspring genotypes. By crossing the gametes from both parents, we get following zygotes. Gametes from parent 1, 8 kind of gametes. Gametes from parent 2, only one kind of gamete. And this is the offspring genotypes. And the fourth step is the third step, sorry, the third step is the phenotype ratios. The most frequent parental type of offsprings are most frequent. Single crossover offsprings are moderate, and double crossover offsprings are least frequent. If we suppose 1,000 offsprings produced, then what will be the number belongs to each type of each kind of phenotype type or genotype? If we add these two parental types, it is equal to the 790. It's mean a parental type of springs is equal to the 790, and single crossover is equal to the, if we add four all four a kind of single crossovers, then it is equal to the 155, and double crossover is only equal to the 55. Our next topic is interference. In other words, we can say that genetic interference. So what is genetic interference? It is a mere how crossovers influence each other during meiosis. In other words, we can say that it is a phenomenon where the occurrence of one crossover event in a specific region of a chromosome reduces the chance of a second crossover event nearby. What is the mechanism? During meiosis, homologous chromosomes exchange genetic material through crossover. If crossover occur close to each other on the same chromosome, they may interfere with one another. There are two types of interference: positive interference and negative interference. In positive interference, there reduces the chances. Positive interference reduces the chances of second crossover occurring near a first one. But negative interference increases the chance of a second crossover near the first one, which is very rare in nature. Interference is measured by the coefficient of coincidence. For the formula of interference is equal to the 1 minus COC. COC mean coefficient of coincidence, and COC is equal to the observed double crossovers over expected double crossovers. Let's have an example. If we have expected crossovers is equal to 10 and observed 7, then COC is equal to the 7 over 10, which is equal to the 0.7. If we put this value in the formula of interference, then 1 minus 0.7 is equal to the 0.3 or 30% interference. This means only 30% less double crossover occurred than the expected due to the interference. What is the biological importance or significance of genetic interference? It ensures the proper chromosome segregation during meiosis, as well as maintains the genetic stability by regulating crossover distribution. What is the difference between linkage map and 3-point test cross? The first difference is linkage map provide a comprehensive representation of gene locations and distances across an entire chromosome or genome. But 3-point test cross analyze a specific set of 3 genes to determine their order and the recombination distances among them. The second difference is that linkage map have broad range, covering many genes across a chromosome or genome. On the other hand, the 3-point test cross has very narrow range, only focused on 3 genes at a time. And the third difference is that linkage map is constructed using multiple test crosses, recombination frequencies, and statistical tools like mapping softwares. But 3-point test cross involves crossing heterozygous and homozygous recessive individuals, analyzing offspring phenotypes, and calculating recombination frequencies manually.